Somewhere inside you, right now, there is a ghost. Not a metaphor, not a poetic flourish, but actual molecular evidence of a group of ancient humans who walked this earth, hunted, survived, loved, and disappeared before anyone alive today could have known their name. They left no written records. They left almost no bones. For most of human history, we had absolutely no idea they had ever existed. And yet their DNA, fragments of their genetic code, lives on inside roughly 5 billion people on Earth today. 5 billion people carrying traces of a human group we only discovered 14 years ago. That is where tonight begins. The group in question are called the Denisovans, and if that name sounds unfamiliar, that's because for most of human history it was. Not just unfamiliar to you or me, unfamiliar to every scientist, every archaeologist, every geneticist on the planet, because the Denisovans weren't discovered the way ancient human groups usually are, through fossils, through skulls, through the slow accumulation of bones in the ground. They were discovered through DNA, a sliver of ancient genetic code pulled from a finger bone so small you could lose it between the cushions of a couch. That discovery upended everything we thought we knew about human prehistory and opened a door into a world far stranger and richer than anyone had imagined. Tonight we're going to walk through that door together. This is a journey through caves and genomes, through ancient migrations and unlikely encounters, through high Tibetan plateaus and tropical islands, and immune systems shaped by pathogens that disappeared thousands of years ago. It's the story of who the Denisovans were, how we found them, and why the traces they left inside the human body still matter right now, in the 21st century, in ways that are genuinely surprising. It's also the story of how science can reconstruct the life of an entire human lineage from almost nothing. No language, no art we can point to with certainty, barely any physical remains. Just fragments of DNA and the extraordinary tools scientists have built to read them. And we're going to do all of this gently. This is a show made for the quiet hours, for the moment when the day has finally let go of you and your mind is starting to drift. You don't need to take notes. You don't need to stay fully awake. The story will carry you where it wants to take you, and if you fall asleep somewhere between Denisova Cave and the ancient islands of Southeast Asia, that is exactly what this was made for. The mystery will still be here when you come back to it. But before we get into the deep history, I want to pause for just a moment and do something I genuinely love. If you're listening to this right now, whether it's midnight in Manila, or early morning in Manchester, or that strange restless 3 a.m. somewhere in the middle of North America, leave a comment and tell me where you are and what time it is. You'd be amazed what those comments look like. It ends up being this strange, beautiful map of the world at rest. People falling asleep on opposite ends of the planet in the same moment, connected by the same quiet curiosity about where we came from. It's one of the best things about making this show, so tell me where you are. I do read them, and if you want to support the work in the most direct way possible, the best thing you can do is follow the show on Spotify. It's ad-free, it's music-free, and it's the version that most directly keeps this going. There's a link in the description. If you've been listening for a while and you've never done that, tonight's a good night for it. Now, back to the ghost. Here is the central mystery that science has been working to unravel for the past decade and a half. Sometime in the deep past, probably more than once, anatomically modern humans encountered another group of ancient humans. Not Neanderthals, though that happened too. A different group entirely. One that had been living across Asia for hundreds of thousands of years before modern humans arrived. And in those encounters, something extraordinary happened. The two groups did not simply compete or avoid each other. They mixed, they interbred, and the genetic legacy of that mixing was passed forward, generation after generation, diluted but never entirely erased, until it reached the 5 billion people alive today who carry some trace of it. What we don't know is almost as interesting as what we do. We don't know exactly what the Denisovans looked like, though some clues are emerging. We don't know how many of them there were, or how far their population extended, though the genetic evidence is beginning to suggest answers that surprised even the scientists who proposed them. We don't know whether modern humans and Denisovans coexisted in the same places for long periods, or whether their encounters were brief and scattered across vast stretches of time and geography. We don't know whether they had language or art or burial rituals, or any of the other cultural markers we usually use to think about human complexity. What we do know is that they were real, that they were human enough to produce fertile offspring with our ancestors, that those offspring survived and had children of their own, and that some of what the Denisovans carried genetically, adaptations shaped by hundreds of thousands of years of living in specific environments, turned out to be genuinely useful to the modern humans who inherited it, in some cases possibly life-saving. That's the thread we're going to follow tonight. From the first hint that something was hidden in ancient DNA, to the cave in Siberia where everything changed, to the genetic map that revealed Denisovan ancestry scattered across the modern world in patterns nobody expected, all the way to what it might actually mean to carry their DNA in your body right now. It's a long journey and a quiet one, the kind best taken at the edge of sleep, when the mind is soft and open and willing to be surprised. So settle in, let your breathing slow. The lights are low, and the story is just beginning. For most of the 20th century, if you opened a textbook on human evolution, you would find a picture. It looks something like a ladder, or maybe a march, a sequence of figures moving from left to right, each one slightly more upright than the last, slightly less hairy, slightly more recognizable as the thing we see in the mirror. Australopithecus shuffling along on the left, then Homo habilis, then Homo erectus, then Neanderthal hunched and heavy-browed, and finally, at the far right, triumphant and vertical, modern humans. The message was implicit but unmistakable. Evolution was a procession, a single file line leading inevitably toward us. It was a compelling picture, clean, directional, satisfying in the way that simple narratives always are. And it was almost entirely wrong. The reality of human evolution, as science has slowly and sometimes reluctantly come to understand, looks nothing like a ladder. It looks more like a tangled delta, a river splitting and rejoining, braiding back through itself, with channels that go dry and others that merge with tributaries from unexpected directions. Multiple groups of humans, not just one, living at the same time in overlapping parts of the world. Not a single procession, but a complicated, messy, deeply human web of populations moving and adapting, and occasionally, crucially, meeting each other. This is the world that existed before the Denisovans had a name, before we even knew there was a gap in our family tree waiting to be filled. To understand what the discovery of the Denisovans actually meant, you have to go back to the way scientists were thinking about human prehistory, even just 20 or 30 years ago. The dominant model was called the recent African origin hypothesis, or sometimes the out-of-Africa theory. The basic idea was that modern humans evolved in Africa somewhere between 200 and 300,000 years ago, and then migrated outward, eventually reaching every corner of the globe. As they spread, they encountered and replaced older human populations that had been living across Europe and Asia for hundreds of thousands of years. Replaced, in most versions of the story, meaning competed with and eventually caused the extinction of. No interbreeding, no blending, just a cleaner, smarter, more capable population moving through and leaving the older groups behind. In this model, those older groups, the ones already living in Eurasia when modern humans arrived, were often lumped together under the umbrella term archaic humans, which is worth pausing on for a moment, because it's a term that still comes up constantly and it can feel more precise than it actually is. Archaic humans doesn't refer to a single species or a single lineage. It's more of a catch-all category for human and human-like populations that predate anatomically modern Homo sapiens, that share some but not all of our features, and that existed in various forms across a huge span of time and geography. Neanderthals fall into this category, so do older populations like Homo heidelbergensis and Homo erectus, and, as we would eventually discover, so do the Denisovans. The problem with treating archaic humans as a single undifferentiated group is that they weren't. They were distinct populations, shaped by hundreds of thousands of years of adaptation to local environments diverging from each other in ways that were real and measurable. Calling them all archaic is a bit like grouping together every animal that isn't a bird and calling them non-avian creatures. Technically accurate, not especially illuminating. But here's the critical thing that the older out-of-Africa model got right, even if it misread the details. Modern humans really did move out of Africa. That much is supported by an enormous convergence of genetic, archaeological and fossil evidence. The migration was real, and when those migrating populations pushed into Europe and Asia, they really did encounter groups that had been there long before them. What the older model got wrong was the idea that those encounters were purely competitive, that modern humans simply swept through and replaced everyone else without leaving any trace of contact in the genome. What was actually happening was considerably more interesting. By the time modern humans began their expansion out of Africa, roughly 60 to 70,000 years ago, Eurasia was already occupied. Not densely, not by modern standards, but occupied in the way that low population density hunter-gatherer societies occupy landscapes. Small groups spread across enormous territories, each group knowing their local environment with extraordinary intimacy, following seasonal resources, moving through the same river valleys and mountain passes generation after generation. Neanderthals had been in Europe and Western Asia for well over 200,000 years. Other populations, the ones we would eventually learn to call Denisovans, had been spread across a vast arc of eastern Eurasia for at least as long, possibly longer. When modern human groups began moving into these territories, contact was probably inevitable, not immediate, not constant. But across the timescales we're talking about, which run into the tens of thousands of years, even rare encounters become statistically certain. Two groups occupying overlapping ranges, competing for similar resources, occasionally finding themselves at the same water source or the same seasonal hunting ground. That kind of contact, stretched across millennia, leaves traces. In the archaeological record sometimes, in the genetic record it turns out, almost always. The trouble was that for most of the 20th century, we didn't have the tools to read those genetic traces. We had bones, we had stone tools, we had careful stratigraphic analysis of sediment layers in caves. These are extraordinary sources of information, and paleoanthropologists have done remarkable things with them. But they have hard limits. A fossil can tell you a lot about the physical form of the creature it came from, the shape of the skull, the density of the bones, the likely size of the brain. It can tell you where and roughly when the creature lived. What it cannot tell you, at least not directly, is how that creature relates genetically to other groups, how much interbreeding may have occurred, or whether the population you're looking at is actually one lineage or several overlapping ones. Those questions required DNA. And for a long time, ancient DNA was simply beyond reach. Genetic material degrades. In warm climates, it degrades fast, often completely, within tens of thousands of years. Even in cooler conditions, the long molecules break down into shorter and shorter fragments, contaminated by bacteria, by the DNA of every person who has ever handled the specimen, by the slow chemistry of time. Extracting useful genetic information from remains that are 50,000 years old, let alone 100,000, seemed for most of the 20th century like a fantasy. And then the tools changed. The development of increasingly precise methods for sequencing ancient DNA transformed paleoanthropology in ways that are genuinely difficult to overstate. Suddenly scientists could ask questions that had previously been unanswerable. Not just who are these remains from, but how does this individual relate to populations living today? How much of their genome ended up in modern humans? Did this group interbreed with that one? How long ago did these two lineages diverge? The bones were the same bones that had always been there. The caves were the same caves. But now, there was an entirely new language available for reading what they contained. This is the context into which the Denisovans were discovered. Not a world of simple answers waiting to be confirmed, but a world of multiplying questions, new tools generating new kinds of evidence, old frameworks crumbling under the weight of data they hadn't been built to accommodate. Scientists already knew, by the early 2000s, that the story of human evolution was more complicated than the ladder model suggested. They were finding evidence of population mixing and divergence in modern human genomes, hints that the out-of-Africa replacement story was too clean, that something messier had happened. But they didn't know what to call the other players in that story. In much of Eurasia, Neanderthals were the obvious candidates. But in Eastern Asia, across the enormous geographic range stretching from Siberia down through Southeast Asia and out into the Pacific, something else had been there. Something whose outlines were only beginning to emerge. The genome was already whispering the answer. In populations across East Asia and Oceania, researchers were finding genetic patterns that didn't fit neatly into any known lineage. Not modern human, not Neanderthal, but something related to both and distinct from both. A signal without a name, a shadow in the data that pointed toward a population nobody had yet identified. That was where things stood. The tools were ready. The question was forming. And somewhere in a cave in the Altai Mountains of Siberia, buried in sediment that had been accumulating for hundreds of thousands of years, a tiny fragment of bone was waiting. The Altai Mountains rise where Russia, Mongolia, Kazakhstan and China lean into each other, a meeting place of continents and climates. In that landscape, where steppe gives way to forest and forest climbs into rock, there is a limestone outcrop with a dark opening in its side. Locals had known it for a long time. They called it Denisova Cave, after a hermit who was said to have lived there centuries ago. To the people who farmed and herded in the surrounding valleys, it was a familiar feature, a place of stories, but not a mystery. To archaeologists, though, caves like Denisova are time machines. Caves do something very simple and very powerful for the archaeological record. They slow everything down. Outside, wind and rain and sun chew at the land, scatter bones, grind stone tools back into dust. Inside, the world is calmer. Sediments fall in through cracks or get tracked in on animal feet and human boots. Dust settles. Ash from ancient fires drifts down. Pollen floats in, gets caught, buried, preserved. Over tens of thousands of years, even a small trickle of material adds up, one grain at a time, into a thick stack of layers. If you cut a careful cross-section through the floor of a cave like Denisova, you don't just see dirt. You see history in vertical profile. The uppermost layers, closest to the surface, tend to be the youngest. The deeper you go, the older things generally become. It's not perfect. Caves flood, roofs collapse, animals dig burrows that mix things up. But in broad strokes, depth is time. Each layer is a page. The whole floor is a book. Archaeologists began working at Denisova Cave in the 1970s. Soviet, then Russian teams sifted the sediments metre by metre, logging every flake of stone, every sliver of bone, every piece of charcoal. Over time they built up an impressive picture of long-term occupation. Humans had been using this cave for a very long time, tens of thousands of years, possibly more than 100,000. They found stone tools, animal remains, traces of hearths. It was, by all appearances, one more important but not world-shattering Paleolithic site in a region already rich with them. The bones, when they turned up, were mostly what you'd expect. Fragments of animal skeletons from hunted prey, bits of rib, shards of long bone, tiny splinters so chewed and broken that you could barely tell what species they once belonged to. Human remains were rare. A tooth here, a bit of bone there. Nothing like the nearly complete skeletons from famous Neanderthal sites in Western Eurasia. And so the cave accumulated data quietly. Layers sampled, artifacts catalogued, sediments bagged and stored for later study. In a sense, the crucial discovery had already happened. It just hadn't been noticed yet. The tiny piece of bone that would change everything surfaced in 2008, during routine sorting of material from one of the deeper layers in the cave's main chamber. To the naked eye, it didn't look like much at all. A scrap of cortical bone from a finger, about the size of a fingernail clipping. No artful curves, no diagnostic features. The kind of thing that, in an earlier era, might have been dismissed as unidentifiable and set aside. Under a microscope, though, the fragment looked human, or at least hominin, one of us, or one of our close cousins. Enough of the internal structure was preserved to suggest it came from the little finger of a juvenile, maybe 8 or 9 years old, a child who had lived and died in or near the cave tens of thousands of years ago. There was nothing in the shape of the bone that screamed new species, no strange proportions, no obvious deformities, no signature Neanderthal thickening. It was just a small, unremarkable piece of a small, unremarkable finger. In the old days, the story would have ended there. A footnote in a site report. Human phalanx fragment from level 11. Probably late Pleistocene. But the days were no longer old. By the late 2000s, ancient DNA work had advanced enough that even very small, very ordinary bones could potentially carry extraordinary information. The team working at Denisova collaborated with geneticists who specialised in extracting DNA from heavily degraded material. This tiny finger bone was sent to one of the leading labs in the world for analysis. To understand what happened next, it helps to think again about those cave layers. Archaeologists had already dated the strata around where the bone was found. They used a combination of techniques, radiocarbon dating of charcoal, which works well up to around 50,000 years, thermoluminescence and optically stimulated luminescence, which measure when mineral grains were last exposed to heat or sunlight. Sometimes uranium series dating, which tracks the slow decay of radioactive elements in calcite deposits. Each method gives you not a single birthday but a range. This layer is probably between X and Y years old, with some margin of error. When you stack these ranges together and cross-check them, a rough but useful timeline emerges. The finger bone came from a level that seemed to date to somewhere around 50 to maybe 70 or 80,000 years ago. Old, but well within the reach of the newest ancient DNA methods. Old enough to be interesting, not so old as to be hopeless. Still, expectations were modest. The most likely outcome, given what was then known, was that the bone would belong to a Neanderthal. Denisova was in Siberia, well within the broad range of Neanderthal populations, and Neanderthal remains had been found at other sites in the wider region. A confirmation of that would have been scientifically valuable. Another Neanderthal genome, another data point. The geneticists drilled a tiny piece from the bone, powdered it, and began the slow, meticulous process of extracting whatever fragments of DNA still clung to the mineral matrix. They filtered out contamination as best they could. They amplified the surviving fragments using modern techniques, then lined them up one by one against reference genomes from living humans and from Neanderthal remains already sequenced. What they saw did not match their expectations. The mitochondrial DNA, the small ring of genetic material inherited only from the mother, was the first clue. It diverged from both modern humans and Neanderthals far more deeply than those two groups diverged from each other. It wasn't just a slightly unusual Neanderthal signal. It was something else entirely, a lineage that had split off from the shared ancestor of Neanderthals and modern humans roughly 1 million years earlier, and that had continued on its own path ever since. That was the first shock. The second came as they sequenced more of the nuclear genome, the main bulk of the DNA housed in the cell nucleus. Once again, the pattern was clear. This was not a modern human. It was not a Neanderthal. It was closely related to both, more closely related to Neanderthals than to us, but distinct from both in ways that were consistent and statistically robust. From this one small fragment of a child's finger, a whole new branch of the human family tree emerged into view. The scientists who published the findings in 2010 were cautious in their language, as scientists tend to be. They spoke of a previously unknown hominin from southern Siberia. They noted that the genetic data suggested a distinct population, one that had shared a common ancestor with Neanderthals hundreds of thousands of years earlier, and then gone its own way. They did not immediately declare a new species. They let the evidence speak. But the effect of that evidence was immediate and global. The unnamed signal that geneticists had been seeing in modern Asian and Oceanian genomes suddenly had a likely source. The shadow in the data had a home, Denisova Cave. The unknown population had a provisional name, Denisovans, the people of Denisova. Almost overnight, a single scrap of bone from a remote Siberian cave turned into a focal point for research groups around the world. Teams began combing museum collections and excavation archives, asking whether any previously discovered remains might actually be Denisovan. New digs were launched or intensified across Siberia, Central Asia, and East Asia, looking for more bones, more teeth, more artifacts in the right time range and the right places. Geneticists reanalyzed existing datasets from modern populations, now with a new reference genome to compare against. The faint mismatched sequences they had been treating as noise suddenly snapped into focus. They weren't noise at all. They were echoes of a real, once-living population that had left its imprint on millions of people alive today. From the outside, it might seem strange that so much could unfold from something so small. From the inside, it made a kind of sense. Caves like Denisova are patient. They collect evidence for hundreds of thousands of years and wait. Technologies change. Questions sharpen. Someone goes back to a box of unsorted bone fragments and sees in a tiny sliver an opportunity. And if the timing is right, if the tools are ready, that tiny sliver can open an entirely new chapter in our understanding of who we are. The Denisovan story began, as far as we know it, with that single piece of bone. But the real power of the discovery lay not just in the fragment itself, but in what it allowed scientists to do next, to read an ancient genome like a time-worn manuscript, and to trace its connections forward into the present. When people talk about ancient DNA, it can sound almost magical, as if scientists are plucking pristine genetic scrolls from the past. The reality is far messier, and in some ways, more impressive. DNA is a fragile molecule. In a living body, it's constantly maintained and repaired, but after death, that maintenance stops. Water seeps into bone and tooth, breaking chemical bonds. Microbes move in, chewing through tissue and genetic material alike. Heat speeds up decay. Oxygen corrodes. Radiation from the environment snips strands into shorter and shorter pieces. Over thousands of years, what was once a long, orderly sequence of 3 billion letters becomes a chaotic cloud of fragments, many only a few dozen letters long. On top of that, old bones don't just contain the DNA of the person who died, they soak up the DNA of everything that touches them. Bacteria, fungi, roots from plants threading through the soil. Then, in the modern era, museum curators, archaeologists and lab workers add their own genetic fingerprints just by handling specimens. A 50,000-year-old bone pulled from a cave floor might at first glance contain far more DNA from the archaeologist who dug it up than from the hominin who once carried it. This is why ancient DNA work looks less like reading a clear page and more like trying to reconstruct a burned book from a handful of ash and a few surviving scraps. Every step is about protecting, isolating, and carefully assembling those scraps, while trying not to mistake someone else's letters for the original text. In the labs that specialise in this, the first enemy is contamination. So those labs are built almost like clean rooms for spacecraft assembly, except the threat runs in the opposite direction. Here, modern DNA is the contaminant, ancient DNA is the delicate cargo. Researchers work in suits, masks, gloves, and sometimes full body coverings. Air is filtered, surfaces are bleached and blasted with ultraviolet light to break down stray DNA. Tools are sterilised between samples. Bone surfaces are shaved away because the outer layers are more likely to have been touched, sneezed on, or invaded by microbes. Often, the people who handle the ancient material are not the same ones who later sequence modern comparison genomes to reduce the risk that their own DNA will quietly sneak into both. Even with those precautions, caution doesn't end when a sequence comes off the machine. Ancient DNA studies are built on replication. The same extract might be processed multiple times. Different pieces of the same bone might be sampled independently. Ideally, separate labs with their own staff, equipment and protocols repeat the work. If one laboratory alone reports a startling new lineage, that's interesting. If a second, working blind with the same material, recovers the same signal, that's much harder to dismiss as error or contamination. This is part of why the Denisovan discovery was taken seriously so quickly. The patterns in that tiny finger bone were checked, rechecked, and cross-checked by different teams. The manuscript was read and reread before anyone dared to say, "This looks like a new chapter." Once the damaged DNA is coaxed out of the bone, another distinction matters: where in the cell that DNA originally came from. Every human cell, like those of other animals, contains two genetic libraries. One is the nuclear genome, housed in the cell nucleus. The long chromosomes, including the fused chromosome that marks our lineage. This is enormous, billions of letters long, and inherited from all of your ancestors, shuffling every generation. The other is mitochondrial DNA, a tiny circular molecule carried in the mitochondria, the cell's energy factories. Mitochondrial DNA is much shorter, only about 16,500 letters, and in humans it's passed down almost exclusively through the maternal line. For ancient DNA work, mitochondrial DNA has one big advantage. There are many more copies of it in each cell. When almost everything has decayed, that redundancy means mitochondrial fragments are often the easiest to recover, especially from very old or poorly preserved material. That's why the mitochondrial genome from the Denisova child was the first part to be read clearly. It's often the first whisper from the past that scientists can hear, but mitochondrial DNA tells only a narrow slice of the story. It can trace maternal lineages, show how deeply one population diverged from another along that female line, and hint at population splits and migrations. It can say, this branch separated from that branch roughly this long ago, at least as far as mothers are concerned. It cannot on its own tell you very much about traits, about interbreeding from other lines, or about the full complexity of a population's ancestry. For that you need the nuclear genome, with its much richer and more tangled record. Nuclear DNA is harder to get from ancient remains. It's less abundant, more fragmented, more likely to have been overwritten by bacterial intruders. When it can be sequenced, though, even in partial form, it opens an entirely different level of resolution. Instead of a single maternal thread, you can begin to see the whole tapestry of ancestry, the contributions from thousands of different forebears, the faint signatures of past interbreeding events. Once enough fragments of nuclear DNA are recovered, they're aligned and compared to reference genomes, those of living humans, Neanderthals, and now Denisovans. Scientists look at where the sequences match closely and where they differ, counting up the number of changes and how they're distributed along the genome. Because mutations accumulate at roughly predictable rates over long timescales, these differences can be used as a kind of molecular clock. It's not an exact clock. Mutation rates can vary somewhat between lineages and over time, and different parts of the genome evolve at different speeds. But with careful calibration and using multiple methods, geneticists can say things like, the common ancestor of Neanderthals and Denisovans probably lived around this long ago with this margin of error. Or, the Denisovan-like DNA in modern populations seems to come from at least 2 separate episodes of interbreeding at different times, with different Denisovan groups. These comparisons also reveal lineages. Clusters of sequences that share distinctive patterns of mutations suggest shared ancestry. When 2 populations share long stretches of identical sequence, it points to recent interbreeding. When they share only short, fragmentary similarities, it hints at deeper, more ancient connections. In this way, genomes become maps. You can trace paths of migration, episodes of isolation, and periods when different human groups met, mixed, and then went their separate ways. But for all their power, these methods come with limits that matter. Ancient DNA survives only in certain conditions. Cold, dry, stable environments are best. Caves help. Permafrost helps. Wet, hot, or acidic soils destroy DNA quickly. That means the genetic record we have is heavily biased toward particular regions and particular kinds of sites. Vast areas of the world, especially in the tropics, have yielded few or no ancient genomes, not because people weren't there, but because their DNA did not last. Even within a site, preservation is uneven. One bone might retain usable DNA, while another, only a few centimetres away, has lost everything. The individuals whose genomes we can read are not a random sample of past populations. They're the ones whose remains happen to be buried in the right place, with the right chemistry, and then happen to be found, recognised and sampled. On top of that, the samples we do have are still few. A handful of bones from a handful of caves, a few dozen genomes, or fragments of genomes, standing in for entire continents and hundreds of thousands of years of history. From that sparse evidence, scientists build models, estimate divergence times, sketch family trees with branches and knots. Those models can be very powerful, especially when they are independently supported by archaeology, by fossils, by climate records. But they are still models, and they can be revised. A single new genome from an unexpected place can redraw entire sections of the map. An additional Denisovan specimen with slightly different genetic features can turn what looked like a single lineage into several. So when we say that Denisovans diverged from Neanderthals at roughly such and such a time, or that a certain percentage of modern genomes carries Denisovan ancestry, those statements rest on layers of assumptions about mutation rates, about which samples are representative, about how many ancient populations have yet to be found. They are our best estimates, not final answers. In that sense, reading ancient DNA really is like working with a time-worn manuscript. The pages are torn, whole chapters are missing. Some lines have been overwritten by later hands. Scholars argue over the right way to reconstruct a sentence, to fill in a gap, to interpret a smudge as one letter or another. But even with all those uncertainties, meaning emerges, patterns appear, a story begins to take shape. The Denisovan child from Denisova Cave is one of those rare, legible pages. Her genome lets us glimpse a population that once ranged across half a continent, and whose descendants still walk the world inside our cells. To understand how those genetic echoes ended up in billions of people today, we have to follow the paths that connected her family to ours, the tangled, knotted branches of a family tree where Neanderthals, Denisovans, and modern humans were never as separate as we once imagined. When people first started drawing human family trees from fossils, the picture looked simple. One branch here, one branch there, all clean splits and tidy names. Homo erectus gives rise to something else, which gives rise to something else, until eventually you get to Neanderthals on one branch, Homo sapiens on another, and for a long time, nothing in between. Denisovans changed that. Their DNA forced everyone to redraw the picture. When geneticists compared the Denisovan genome to that of Neanderthals and modern humans, a pattern emerged. Denisovans and Neanderthals were closer to each other than either was to us. In very broad strokes, it looks like this. First, There was a population of ancient humans that had already passed through that great bottleneck. Sometime after the population exploded out of Africa into Eurasia, one branch split off that would eventually lead to Neanderthals and Denisovans. Another, still in Africa, would keep evolving toward what we call Homo sapiens. Then, within that Eurasian branch, another split. One line moving toward the Neanderthals we know from Europe and Western Asia, and another line heading toward the Denisovans, who would occupy much of Asia and its island fringes. If you compress hundreds of thousands of years into a few lines on a page, that's what you get. A neat little fork, then another, 3 labelled tips. Modern humans, Neanderthals, Denisovans. 3 sister groups, each more closely related to one another than to more distant branches like Homo erectus. In genetics, a sister group just means 2 lineages that share a more recent common ancestor with each other than either does with anyone else. Neanderthals and Denisovans are sister groups because they split from each other after their shared line had already split from ours. In a purely branching tree, you could put a dot for that ancestor, draw 2 lines, and be done. But the more DNA we read, the less our history looks like a tidy tree, and the more it looks like a web. For one thing, those splits are not sharp, single moments in time. Populations don't wake up one morning and decide to become separate species. What happens instead is that groups drift apart. They move into different regions, adapt to different climates, accumulate different mutations. Over thousands of generations, gene flow between them becomes rarer and rarer. There might be centuries when they scarcely meet, followed by a brief period of contact where a few individuals mix and exchange genes, and then separation again. If you could watch all of this in real time, you wouldn't see crisp branching. You'd see fuzzy edges, strands peeling apart and then briefly rejoining, braiding and unbraiding before finally drifting far enough apart that contact becomes unlikely. The Denisovan story is a perfect example. When the first Denisovan genome was published, it was tempting to talk about the Denisovans as if they were a single coherent population, one people, with one territory, diverging from Neanderthals at one approximate date. But the signals in modern DNA tell a more complicated story. When researchers look at Denisovan-like sequences in living populations, they don't all match the Denisova cave genome equally well. Denisovan ancestry in mainland Asia, in New Guinea, in Australia, in the Philippines, and in parts of East Asia seems to come from at least 2, and probably more, distinct Denisovan lineages. Some are more closely related to that Siberian child's group. Others look like cousins that had split off long before, evolving on their own, perhaps thousands of miles away. In other words, Denisovan is not one branch. It's more like a whole cluster of branches, some of them fairly close to Neanderthals, others more distant, all sharing a deep ancestor but spending a long time apart. And Denisovans weren't alone in this. Neanderthals themselves were not a single uniform population. Genomes from different Neanderthal sites hint at regional subgroups, waves of expansion and replacement, periods of isolation followed by contact. Even within Africa, where our own lineage was taking shape, the genetic evidence points to structured populations, semi-isolated groups with their own histories, occasionally mixing, occasionally drifting apart. If you try to draw all of that on a piece of paper, the lines start to cross. A Neanderthal-like population might branch from a common ancestor, spread west, then send a small offshoot east that mixes with an early Denisovan-like group. Later, another wave moves back through the same region and mixes again, adding new genes and diluting old ones. Meanwhile, some Denisovan line splits, one branch moving into Southeast Asia and interbreeding with the ancestors of modern Papuans, another staying somewhere farther north, leaving traces in East Asian genomes. Each of those events ties a knot in the tree. A line that looked separate suddenly loops back and connects to another. The branches are still there, but they're stitched together by threads of gene flow. This is why geneticists often talk about admixture instead of clean splits, and why they use models that allow for multiple pulses of interbreeding between partially separated groups. It's also why broad timelines are given with caution. We can estimate when the ancestor of Neanderthals and Denisovans diverged from the ancestor of modern humans. We can estimate when Neanderthals and Denisovans themselves started becoming distinct. But those dates are approximations, averages over long, messy processes, not single birthdays for new species. The names we use can hide that messiness. Neanderthal conjures a particular image: stocky limbs, European caves, Ice Age hunts. Denisovan now evokes Siberian caves, island shores, high mountain plateaus. But under each label there were likely many regional groups, each with its own dialects, tools, alliances and genetic quirks, living over vast stretches of time. The Denisovans, who left DNA in the ancestors of Tibetan highlanders, may not have seen themselves as the same people as the Denisovans who left DNA in the ancestors of New Guineans. They might never have met at all. From our distance, with only a handful of bones and sequences, we compress all that diversity into a single word. That's useful for talking about patterns. It's less good at capturing reality on the ground, where boundaries were soft, identities were local, and genes were always on the move. So when we picture Neanderthals, Denisovans, and modern humans, it might be better not to imagine three separate tribes marching along their own paths. Instead, imagine a landscape full of related bands, spreading and shrinking, sometimes isolated by mountains or deserts or ice sheets, sometimes reconnecting along rivers and coasts and mountain passes. A landscape where sister groups are real, but the lines between them are blurred, and where every so often two distant cousins meet, have children, and quietly tie another knot in the family web. Those knots matter, because they're how Denisovan DNA slipped into our own line, how genes from far-flung, now-vanished populations ended up in billions of people today. To see how those encounters might have happened, we need to leave the clean diagrams behind and step into the actual world those humans walked, a world of shifting ranges, chance meetings, and shared nights by unfamiliar fires. As modern humans began pushing out of Africa for good, that web of related bands became a world of encounters. The first big contact zone was probably the corridor stretching from the eastern Mediterranean through the Levant and into Western Asia. For hundreds of thousands of years, Neanderthal-like populations had used this region as a stepping stone, moving south during cold phases and retreating north when it warmed. When our ancestors finally came through in larger numbers, they weren't walking into empty land. They were entering a space that had been occupied, abandoned and reoccupied many times by other humans. Genetic studies suggest that some of the earliest mixing between Homo sapiens and Neanderthals happened here. Small groups of our ancestors, following rivers and coastlines, would have met Neanderthal bands using the same valleys, the same springs, the same herds. Some meetings may have ended in wary avoidance, each group watching the other from a distance, trying to decide if these strange familiar creatures were competitors, trade partners, or something in between. Other meetings, statistically, would have gone further. From there, as modern humans moved east along the southern arc of Asia, new contact zones opened. The coasts of India, the river systems of Southeast Asia, the mountain foothills of what is now China. Neanderthals seem to have pushed farther east than once thought, and Denisovan-like populations probably occupied much of the interior and the highlands. The genetic signals we see today suggest that as our ancestors threaded their way through these landscapes, they didn't meet Denisovans just once. They met different Denisovan groups at different times in different places. Think of the Arabian Peninsula, the coasts of the Indian Ocean, and the lowland forests and river valleys of Southeast Asia as overlapping territories. Modern humans expanding along one route, Denisovan-like populations holding ground along another. Where those ranges overlapped, even briefly, you get opportunities for contact. A band camping near a rich fishing spot might find that another band is already using it. At first there might be tension, posturing, maybe even violence. But resources shift, seasons change, and sometimes, instead of one group displacing the other, you get an uneasy truce. Those are exactly the conditions under which introgression happens. Introgression is the term geneticists use for the process by which small amounts of DNA from one population or species enter the gene pool of another, and then either disappear or stick around. At the individual level, it's simple. Two people from different groups have a child. That child carries a mixture of DNA from both. If their descendants keep marrying mostly within one group, the DNA from the other population gets diluted each generation. But some segments can hang on, especially if they happen to help with survival. Imagine a Denisovan woman and a modern human man forming a pair within a small coastal band. Their children might be raised speaking the language of the majority, using tools from that cultural tradition, thinking of themselves as part of that group. Genetically, though, they carry a little Denisovan DNA. If life is hard and only a few children survive, chance alone might erase that Denisovan contribution in a few generations. One unlucky accident, one epidemic, and those lineages vanish. But if those children survive and have children of their own, and if some of the Denisovan-derived variants happen to improve resistance to a local disease, or make high-altitude breathing easier, or help digest a new kind of food, then the odds change. Individuals carrying those segments are more likely on average to live long enough to have children. Over hundreds of generations, those helpful fragments can spread, even as everything else from that ancient encounter fades away. This is why interbreeding doesn't require huge numbers of mixed families to leave a mark. You don't need a continent-wide blending of populations. You just need a handful of successful unions in the right places, repeated here and there over thousands of years. Each one is a tiny experiment in genetic exchange. Most leave little trace. A few leave echoes that can still be read in billions of people. What did those encounters look like up close? The genomes can't tell us directly, but they allow us to sketch the range of possibilities. Some contact zones may have been mostly hostile. Bands competing for the same herds or the same waterholes might have driven each other off, raided each other's camps, or simply kept as much distance as possible. In such regions, mixing would have been rare and often one-sided, driven more by capture and coercion than by cooperation. The DNA alone can't distinguish between a consensual partnership and a forced one. Other regions may have seen periods of coexistence. Two bands camped on opposite sides of a valley, watching each other's fires at night, occasionally approaching to exchange meat for stone, shells for pigments, stories for news of distant places. In small populations, any long-term alliance like that becomes entangled with family. A trade partner becomes a son-in-law. A visiting healer becomes a grandmother. Over time, kinship ties could bind previously separate groups into a loose network, with genes flowing along the same routes as obsidian and ornaments. There might also have been truly mixed communities, at least for a time, places where individuals with partly Neanderthal ancestry, partly Denisovan ancestry, and mostly modern human ancestry all lived together, sharing tools and myths, arguing and laughing, raising children who carried several strands of the human web inside them. In such bands, species labels lose their meaning. What you notice isn't cranial shape or nasal width. It's who knows the migration routes of the herds, who remembers where to find edible roots in the dry season, who has relatives in the next valley. Genomes give us a few solid anchors in this sea of possibility. They tell us that there were multiple pulses of gene flow from Neanderthals into modern humans, not just one. They tell us that Denisovan-like DNA entered our lineage more than once, from at least two distinct Denisovan populations. They tell us that some of these events happened far to the east, near the ancestors of present-day Papuans and Australians, and others closer to East Asia. They reveal that the Denisovan and Neanderthal lineages themselves sometimes mixed, long before either met our branch. But beyond timing, direction, and rough location, the genomes fall quiet. They can't tell us the words spoken at that first meeting by a cold river. They can't tell us whether those encounters were framed as marriage alliances, peace offerings, or desperate gambles in hard times. They can't tell us what names people used for themselves, or whether they drew mental lines between us and them in ways that match the categories we use now. What they do show is that whatever those lines were, they were permeable. Again and again, in different corners of Eurasia, members of these related but distinct human groups crossed them. Not often, relative to all the births and deaths that filled those millennia, but often enough that their choices are written into our cells. The result is a world where modern humans carry Neanderthal and Denisovan genes without ever having heard of either name until very recently. A world where ancient encounters in forgotten valleys still shape how we breathe, fight infection, and respond to our environments. And a world where, to trace the path of Denisovan DNA today, we have to follow the routes those mixed bands took as they fanned out across Asia and beyond. If you could fall upward, out of your own body, and into a kind of genetic night sky, and look down at the world as nothing but patterns of ancestry, the Denisovan signal wouldn't be spread evenly like moonlight. It would be patchy, bright in some regions, faint glimmers in others, and almost invisible over wide stretches of land. And yet, when you step back far enough, you start to see something astonishing. Those faint glimmers add up to a glow that touches billions of people. The clearest, brightest Denisovan glow today is not over the great continents of Eurasia, but over the islands strung across the Western Pacific. In places like New Guinea, parts of island Southeast Asia, and among many indigenous Australian groups, people carry the highest known proportions of Denisovan ancestry in the world. Not a majority of their genomes, not even close, but several percent. When you're talking about billions of letters of DNA, a few percent is a lot. It's the difference between a barely audible whisper and a steady background hum. On a map, if you shade regions based on average Denisovan ancestry, the darkest colours settle over New Guinea, over island chains like the Bismarcks and the Solomons, over parts of eastern Indonesia and the surrounding seas. Here, the echo of those mysterious eastern Denisovan populations is strongest. The ancestors of these island peoples seem to have passed through contact zones where Denisovans were still common, or at least common enough for several episodes of mixing to leave a deep imprint. Move north and west from there, and the colours start to fade, but they don't disappear. In mainland Southeast Asia, in southern China, in parts of East Asia, Denisovan ancestry is lower on average, but still detectable. The percentages may drop from several percent to less than 1%, but remember what that actually means. Even 0.2% Denisovan in a modern genome still represents millions of letters of code passed down across tens of thousands of years. One way to picture it is like a mural that's been painted over again and again. In Oceania, large patches of the original Denisovan paint are still visible through the later layers. In much of Asia, only small flecks remain. You could stand in front of the wall and not notice them at first. But if you lean in, if you shine the right light, they're there. Tiny, scattered, but real. This is where the number 5 billion comes from. It doesn't mean that 5 billion people are walking around with the same hefty dose of Denisovan ancestry we see in Papuan or indigenous Australian populations. It means that if you take a broad look across the globe and you add up all the small scattered fragments of Denisovan-derived DNA in all the different populations where genetic studies have found them, the number of people carrying at least a little of that legacy could be on the order of half the human species. In some people, that contribution is concentrated. In others, it's like a dusting of snow on a distant mountaintop, almost subliminal. But at the scale of billions, even very modest averages translate into huge numbers of individuals. East and South Asia are where the map becomes especially strange, and where the story becomes much more tangled than a simple more here, less there picture. When geneticists first looked for Denisovan DNA in Asians, they expected a smooth gradient. The closer to Denisova cave, the more Denisovan ancestry. Instead, they found something more like knots and eddies in a river. Some East Asian populations showed signs of Denisovan-like DNA that didn't match the Denisovan signals seen in Papuans and Australians. Others showed little or none. Some South Asian groups carried subtle Denisovan-like fragments, while nearby groups seemed to lack them, or carry different ones. The simplest way to explain this is that our ancestors didn't just meet one Denisovan population, at one time, in one place. They seem to have met at least two, maybe more, and at different points along their journeys. One branch of Denisovan-like humans may have mixed with the ancestors of present-day Papuans and Australians somewhere in Southeast Asia. Another, genetically distinct Denisovan group may have mixed with populations closer to East Asia. Later migrations and population shifts then layered new waves of ancestry on top of the old, sometimes diluting those ancient segments, sometimes preserving them in isolated pockets. From a distance, all of this complexity flattens into averages. You see a number, 0.3% here, 0.8% there. But at the level of individual people, it's far more uneven. One person in a village might carry several long Denisovan-derived segments, another almost none. Over many individuals, the average smooths out. But the route that DNA took to get there was anything but smooth. And even the maps we have now are not final. The picture of Denisovan ancestry that you see in textbooks or news articles is based on the populations that have been sampled so far. That sounds obvious, but it has enormous consequences. For a long time, almost all the genome data came from Europe, East Asia, and a few places in the Americas. Whole regions of the world, especially in Africa and parts of Asia and the Pacific, were barely represented. When researchers started sequencing genomes from indigenous groups in New Guinea and nearby islands, estimates of Denisovan ancestry in those regions jumped. Not because Denisovan DNA had suddenly increased, but because we had finally looked. As more indigenous communities choose to participate in research, and as geneticists improve their methods, the map keeps changing. Sometimes an area that once looked blank becomes filled with subtle Denisovan signals. Sometimes a region that seemed to have high Denisovan ancestry turns out to be more complicated, with overlapping layers of Neanderthal-derived and Denisovan-derived segments that earlier tools couldn't easily separate. In some cases, what looked like a neat boundary on the map dissolves into a gradient once more samples are added. This is another reason to be cautious with neat numbers and sharp lines. When you see a figure like 2% Denisovan here, 0.1% there, you're not looking at a timeless truth. You're looking at a snapshot of our current data and our current methods. Add more people, especially from undersampled regions. Refine the statistical models. The colours on the map can shift. And yet, through all those updates, one thing has become clearer rather than fuzzier. Denisovan ancestry isn't a curious regional quirk confined to a few remote islands. It's part of a much larger interconnected pattern, strong in Oceania, complex and layered in East and South Asia, faint but present in other parts of the world. A residue of ancient meetings spread out across such a large fraction of humanity that it's more surprising not to carry any at all. To understand how those strong pockets in the Pacific formed, and how those faint traces ended up scattered across so much of Asia, we have to follow our ancestors as they did something bold and new. They learned to cross open water. During the Ice Age, the world your ancestors walked through didn't look quite like the one on a modern atlas. Sea levels were lower, locked up in vast ice sheets. Coastlines were pushed outward. Shallow seas became exposed as broad, humid plains. In Southeast Asia, two hidden continents appeared. To the west was Sunda, a drowned landmass that once connected what are now the Malay Peninsula, Borneo, Java, Sumatra, and many of the islands in between. When the seas fell, you could, in principle, have walked on dry ground from what is now mainland Southeast Asia all the way to the edge of what we'd recognise as Indonesia, weaving through river valleys and low hills instead of island hopping across blue water. Far to the east was another lost continent, Sahul. This was not just Australia. It was Australia fused with New Guinea and Tasmania into a single, huge landmass, ringed by reefs and shallow shelves, home to marsupials, giant flightless birds, and forests and grasslands that no human eye had ever seen before our species arrived. Between Sunda and Sahul lay something that never disappeared, even at the height of the cold: water, deep channels, stretches of sea that did not dry out when the climate froze, a broken chain of islands and island ridges, yes, but no continuous bridge. This region is sometimes called Wallacea, after the 19th century naturalist Alfred Russel Wallace, who noticed that animal species changed abruptly as you crossed from the Asian side to the Australian side, monkeys and tigers on one side, marsupials and strange birds on the other. The line between them was not a fence of rock, it was salt water. To reach Sahul from Sunda, your ancestors could not just follow herds over a land bridge. At some point in some direction they had to face a horizon that did not show any solid ground at all. They had to cross open water. Now, exactly how that happened is still being debated. No one was there with a notebook, but the geography sets limits on what is possible. Even if you imagine the most favourable scenario, calm seas, low tides, clear weather, the distances between some of these islands were too great to swim, especially if you were carrying children, tools, food, and the hopes of your band. People could have waded along shorelines, floated on logs, clung to natural rafts of vegetation torn from riverbanks by floods. That might work for a single crossing. But to move a whole population step by step from Sunda to Sahul, you need something more than luck. You need planning. Maybe it began simply: a log that drifted, a group that paddled with their hands, a few curious individuals who pushed out of sight of shore because they saw a darker smudge on the horizon. But somewhere along this chain of crossings, people had to learn which direction to go when the tides were favourable, which winds helped and which were deadly. They had to experiment, fail, try again, remember, teach. That's what boat really means here. Not necessarily a sleek canoe with a carved prow, but an organised way of thinking about water travel. Tying logs together, shaping wood to float better, using bundled reeds, bark, or skins. Knowing that if you push off from this bay at this time of year and keep the setting sun on your left shoulder, an island will be there when the stars come out. Some archaeologists think these journeys to Sahul might have been accidental, a one-time drift of a few families blown hopelessly off course. Others argue that the numbers don't work. To found a population in Sahul large enough to survive and grow, you probably need multiple crossings, with dozens or even hundreds of people over time. That looks less like an accident and more like a strategy. And however they did it, They didn't do it through empty lands. Wallacea and the surrounding islands weren't a blank slate waiting politely for modern humans to arrive. The genetic signals we see today, and faint hints from stone tools and scattered fossils, point to something much messier, a patchwork of archaic populations already living there. Denisovan-related groups are the prime suspects for at least some of these island dwellers. The high levels of Denisovan ancestry in Papuans and indigenous Australians suggest that, somewhere in this broad island world, the ancestors of Sahul's first peoples met and mixed with Denisovan-like humans before or during the crossing. The timing isn't nailed down, the exact islands are unknown, but the pattern in the DNA implies contact zones, not a single handshake on a single beach, but repeated interactions as modern human groups move through a landscape already occupied. Island Southeast Asia is particularly good at preserving complexity. Its geography helps. You can imagine it as a maze of stepping stones, with channels of water serving both as barriers and filters. A group of archaic humans might adapt to the forests and mountains of one large island and, for tens of thousands of years, rarely if ever cross the water gap to the next one. Another group, slightly more adventurous or better at building rafts, might spread along a chain of smaller islands, interbreeding occasionally with their neighbours, but never quite blending into a single continuous population. Sea levels rise and fall, coastlines advance and retreat, islands merge into peninsulas and then fragment again. Each change cuts off some groups, reconnects others, and reshapes the map of who can meet whom. Over long stretches of time, this kind of shifting archipelago can act like a laboratory for evolution, keeping lineages isolated just long enough to drift apart, then bringing them back into contact. It's easy, in this region, to imagine more than one archaic population surviving side by side. Maybe there were Denisovan-like groups adapted to lowland rainforests, and others better suited to the drier savannas that appeared during cold phases. Maybe a cousin population, related but distinct, clung to high volcanic slopes. When modern humans entered this world of islands and narrow seas, they would not have found a single Denisovan territory, but a mosaic of homelands, some already abandoned, some very much alive. This is part of why the question, where did Denisovans live, is still so hard to answer. The only place where we can point to a cave and say, with confidence, Denisovans were here, is in the Altai Mountains of Siberia. Yet the strongest Denisovan genetic footprints today are thousands of kilometres away, in populations that trace their deep roots to New Guinea, Australia and nearby islands. So did Denisovans range all the way from Siberia to island Southeast Asia? Did they exist as a single widespread species, flowing across Asia and splitting into regional branches? Or are we using one fossil and one genome from Denisova Cave as a stand-in for a whole family of related archaic groups that may have been diverse, scattered, and only loosely connected? Different researchers lean in different directions. Some argue for a wide Denisovan range, with local populations adapting to high mountains here, tropical islands there, and cold northern forests somewhere in between. Others suggest that what we call Denisovan might actually hide multiple distinct lineages, some of which we've only glimpsed through their echoes in modern DNA. For now, the islands hold more questions than answers. We don't yet know which specific channel crossings were the ones where Denisovan and modern human families first shared food, stories and genes. We don't know which island ridges sheltered Denisovan groups for the longest, or where their final refuges may have been as sea levels rose and new waves of people arrived. What we do know is that by the time the first humans stood on the shores of Sahul and looked inland at a continent no human had ever seen, they were already carrying more than their own history. In their cells, knotted into their chromosomes, they carried fragments of island encounters, pieces of DNA that had crossed seas long before anyone drew a map. Some of those fragments would turn out to be more than just curiosities. In certain places, Denisovan-derived genes seem to have done something remarkable. They helped our ancestors survive in environments that would otherwise have been deadly. One of the clearest examples lies far from the sea, high above the clouds, on the roof of the world. High altitude sounds romantic from far away. Snow peaks, thin blue air, a roof of the world sunrise above the clouds. But for a body that evolved at sea level, going up fast is more like an emergency. The air doesn't feel that different in your lungs. You can still pull it in, still fill your chest. The problem is more subtle. At 3, 4, 5,000 metres above sea level, Every breath carries fewer oxygen molecules. The pressure is lower, the air is thinner, and the whole system your ancestors built over millions of years to move oxygen from the outside world into your cells starts to struggle. If you or I, unadapted, climb quickly to those heights, our bodies panic. Heart rate spikes. Breathing speeds up. Blood vessels in the lungs constrict in strange ways. Headaches, nausea, dizziness. In serious cases, fluid can leak into the brain or the lungs, a deadly condition called high-altitude cerebral or pulmonary edema. Even healthy young adults can be killed by the climb if they go too fast or stay too long. And yet, on the Tibetan Plateau, on the ridgelines of the Himalaya, in villages perched where lowlanders would gasp for air, millions of people live and work and raise children as if this were normal. For them, it is. Their bodies do something unusual in the thin air. Instead of responding by cranking up red blood cell production to extreme levels, which thickens the blood and can cause its own problems, Tibetan highlanders seem to hold a different balance. They adjust breathing and circulation, but they don't push their blood chemistry into the dangerous territory that many lowlanders reach at altitude. This isn't a matter of just getting used to the mountains. It's written, at least partly, in their genes. When geneticists began comparing Tibetan populations to nearby lowland groups, one difference leapt out again and again, a stretch of DNA near a gene called EPAS1. EPAS1 is not a high-altitude gene, in the sense of being switched off at sea level and switched on in the mountains. It's part of a general system your body uses all the time, a kind of oxygen-sensing alarm network. Cells constantly monitor how much oxygen they have, and EPAS1 helps regulate the response, how blood vessels behave, how much hemoglobin you make, how your body copes when oxygen runs low. In most human groups, EPAS1 comes in a few common versions, but among Tibetans, one particular version is unusually frequent, especially in people whose families have lived at high altitude for many generations. It's associated with lower hemoglobin levels than you'd expect at those heights, low compared to other highlanders, but just right for avoiding the dangers of sludge-thick blood. When researchers looked more closely at this Tibetan EPAS1 variant, they found something odd. It didn't look like the typical global human pattern. Its sequence was different. Not just a little different, but different in ways that statistically are hard to explain if you assume it evolved entirely within the line of modern humans after we left Africa. So they asked another question. If this variant doesn't look like regular modern human DNA, does it look like anyone else's? That's when the Denisovans walked back into the story. When scientists compared the Tibetan EPAS1 sequence to ancient genomes, they found a striking match with the DNA from Denisovans. Not identical in every letter, but close enough that the simplest explanation is that this chunk of DNA entered the ancestors of Tibetans through interbreeding with a Denisovan-related population somewhere in their past. In other words, a piece of Denisovan oxygen response machinery appears to have been borrowed, carried forward, and then sharpened by natural selection in high-altitude humans. This is what geneticists call introgression, genes moving from one population into another through mating, then being passed down the generations. Most introgressed DNA doesn't do anything dramatic. It just drifts along, occasionally nudged by selection, often slowly diluted. But every once in a while, a borrowed variant lands in a context where it's unbelievably useful. Imagine a small group of modern humans moving into the uplands of Central Asia, or into the rising shadow of the Himalaya. Some of them carry a Denisovan-derived EPAS1 variant, picked up generations earlier from contact with archaic populations somewhere in Asia. At sea level, this variant doesn't make much difference. Maybe it's neutral, maybe it has a tiny cost, it just sits there. At altitude, it becomes a lifeline. Children who inherit this version of EPAS1 can cope better with hypoxia. They avoid some of the worst pregnancy complications that high altitude can cause. Their hearts and lungs are less strained. They're a little more likely to survive infancy, a little more likely to grow up and have children of their own. Natural selection doesn't need a giant advantage to work. It just needs consistent tiny differences in survival and reproduction, generation after generation. Over thousands of years on the Tibetan Plateau, the EPAS1 variant that helped people live and have healthy babies in thin air rose in frequency, like a favourable current carrying a particular kind of boat downstream. Today, that Denisovan-like EPAS1 signature is among the strongest signals of archaic introgression we can see anywhere in the human genome. It's not a ghostly trace of ancient history. It's doing something right now, in the cells of people breathing the cold, high air above the tree line. There are still debates, and they matter. We don't yet know exactly where this Denisovan-related EPAS1 variant first entered the ancestors of Tibetan highlanders. Was it picked up far to the east, in the mountains of what is now western China? Was it introduced earlier, in lowland populations, and only later favoured when some of their descendants climbed into higher elevations? Or did it come from a Denisovan-like group adapted to mountains somewhere else entirely, perhaps in Central Asia, whose bones we haven't yet found? We also don't know precisely when the key introgression happened. Genetic estimates suggest that the variant may have been present in broader Asian populations long before humans occupied the Tibetan plateau in large numbers. The selection that made it common among Tibetans could be relatively recent on evolutionary timescales, tens of thousands of years, not hundreds. And we don't know which Denisovan population was involved. The Denisovan genome we have from Siberia is just one data point. Other Denisovan-like groups, genetically distinct, could have been living closer to the Tibetan plateau, even on it. Some researchers suspect that the EPAS1-like sequence may come from such a regional Denisovan population, one more specialised for life at altitude than the Siberian group we've sampled. What is solid is the core of the story. There is a specific, functionally important region of the genome that looks Denisovan in origin and appears to have been strongly favoured by natural selection in Tibetan highlanders. Its effect on hemoglobin regulation and oxygen handling fits with what we see physiologically, and its frequency pattern, common in Tibetans, much rarer in neighbouring lowland groups, matches what you'd expect if high altitude itself had been the selective force. This matters for more than local adaptation. It changes how we think about archaic DNA in all of us. It's easy to treat Neanderthal and Denisovan sequences as a kind of genetic museum. Ancient artefacts locked in our chromosomes, interesting but inert. The EPAS1 story shows another side. Archaic DNA can be machinery, not just memory. It can be a tool that natural selection picks up and refines, turning an old solution to one environment into a new solution to another. On the Tibetan plateau, that machinery hums quietly in the background of every breath. A molecular reminder that the boundaries between our species and theirs were never simple walls. They were more like semi-permeable membranes, letting useful pieces through, then letting time and environment decide which one stayed. As you picture the high villages under the stars, prayer flags snapping in winds that smell of ice and stone, you can imagine more than just human endurance. Somewhere far beneath the skin, in the pattern of bases along a strand of DNA, a Denisovan inheritance is still doing its work, helping hearts beat steadily in air that would leave most of us gasping. And EPAS1 is not alone. In other corners of the genome, and in other parts of the world, archaic DNA has left traces that shaped how our immune systems work, how we handle cold, how we respond to unfamiliar pathogens. The mountains give us a clear example. To see the others, we have to look inward, into the invisible war everybody fights, every day, against the microscopic world. If high altitude is a slow, suffocating pressure on the body, then pathogens are something else entirely. They are ambushes. You can walk through a forest, wade a river, share a shelter with strangers, and never see the things most likely to kill you. Bacteria on a cut, a virus in a cough, a parasite in the water from a quiet, clear stream. For most of human history, this invisible world set the rules. Your ancestors didn't know about microbes, but their bodies acted as if they did. Every cut that didn't fester, every fever that broke instead of spiralling out of control, every child who survived a mysterious sickness instead of being buried under a cairn of stones. All of that left its faint mark on the genomes of the people who lived. That's why genes involved in immunity so often look disturbed. Compared to many other parts of the genome, immune-related regions are full of scars, odd patterns of variation, sharp differences between populations, stretches that seem to have changed unusually fast. Geneticists call this a signature of selection. It's what you see when something is being tested, again and again, against a shifting enemy. Imagine the immune system as a library of molecular wanted posters. Each poster recognizes a pattern: a viral shell, a bacterial toxin, a parasite surface protein. Pathogens evolve to change those patterns, to slip past recognition. The host evolves to update the posters, to notice the new disguises. Back and forth, over millions of years. The arms race doesn't stop when a new human species appears, and it doesn't stop when different humans meet. When modern humans moved into Eurasia, they didn't just encounter Neanderthals and Denisovans. They encountered Neanderthal and Denisovan pathogens, local parasites, endemic viruses, long-established bacterial communities in soils, waters, and animal reservoirs. For the archaic populations, these microbes were familiar opponents. For the newcomers, they were fresh threats. In that situation, introgressed DNA could act like a borrowed field guide. If a modern human and a Denisovan had children, some of those children would inherit Denisovan variants in genes that helped recognize or respond to local pathogens. Things like receptors that sit on immune cells and scan for danger, or proteins that switch on inflammatory responses when something looks wrong. In the abstract, they're just little differences in sequence. But in practice, they can be the difference between dying from a first exposure or walking away with a new scar and a stronger immune memory. Several gene regions often come up when researchers talk about this kind of archaic contribution. One group is the toll-like receptors, especially genes like TLR1, TLR6, and TLR10. These are part of the innate immune system, the fast, blunt, first-line response that doesn't wait for a custom antibody. Some variants in these regions in modern Eurasian populations seem to carry a clear Neanderthal or Denisovan signature. They may have tuned the sensitivity of early pathogen detection, making cells more or less likely to raise the alarm when they encountered particular microbial patterns. Another frequent topic is the HLA region, the human leukocyte antigen system. This is one of the most diverse parts of the genome, because it handles how your body presents bits of pathogens to immune cells. A wider repertoire of HLA variants means your population, collectively, can recognize a wider array of threats. A number of HLA types in modern people appear to trace back, at least in part, to archaic introgression. There are others, components of antiviral defense pathways, genes involved in interacting with RNA viruses, elements of the complement system that helps tag and clear invaders. In many of these places, archaic-like haplotypes sit among the branches of the modern human family tree, suggesting that when we met Neanderthals and Denisovans, we didn't just exchange faces and bone structure, we exchanged immunological tricks. But this isn't a simple story of good, immune genes from ancient cousins. The same changes that help in one context can hurt in another. Dial the innate immune system up, and you might get better protection against certain infections. You might also get more collateral damage, chronic inflammation, greater risk of autoimmune reactions where the body mistakes its own tissues for the enemy. Tune HLA presentation in one direction, and you may become better at recognizing some parasites but slightly worse at handling others. A variant that helped your ancestors survive a lethal outbreak 10,000 years ago could, in a different environment, quietly raise the odds of allergy, asthma, or other inflammatory conditions. Researchers see hints of these trade-offs when they look at modern health data. Some introgressed immune variants are statistically associated with differences in susceptibility to infections, or with autoimmune and inflammatory diseases. But those links are often weak and easily tangled with lifestyle, environment, and countless other genetic factors. The truth is it's complicated. An immune gene is not a light switch. It is a knob in a vast control board of pathways and feedback loops. A Denisovan-derived variant in one of those knobs might change the timing or strength of a response by a small amount. Whether that's helpful or harmful can depend on diet, climate, exposure to farm animals, sanitation, even the kinds of childhood infections common in a particular place and time. And then there's the historical layering. Modern people with Denisovan ancestry are not living in Denisovan environments. The diseases of ancient Pleistocene Asia are not the same as those circulating in modern megacities or agricultural valleys. A variant that once balanced the risks of gut parasites and wound infections in a foraging society will interact very differently with antibiotics, vaccines, and dense urban living. So geneticists are cautious. They can say with some confidence that immune-related genes show some of the clearest signs of selection on archaic DNA. They can point to specific regions where Neanderthal or Denisovan-like sequences seem to have been favoured in non-African populations, likely because they provided an advantage against local pathogens. They can map where those haplotypes are common today. What they cannot yet do in most cases is trace a simple, direct line from a particular archaic variant to a specific modern medical outcome and say, this causes that. Instead, they talk in probabilities and tendencies, in small shifts in risk and resilience layered over many generations. They build models, compare populations, test hypotheses in cells and in animals, and slowly refine the picture. Amid all that complexity, one idea remains clear. When our ancestors met Denisovans and Neanderthals, they didn't just acquire stories and children, they acquired pieces of an immune system that had already spent hundreds of thousands of years wrestling with Eurasian microbes. Those pieces were then reshaped by selection in our own line, woven into our biology, and carried forward into lives utterly unlike those of the people who first contributed them. Somewhere in the quiet workings of your own immune cells, in how your body reacts to a scratch, a flu virus, a gut bug, there may be traces of that ancient exchange. Not a magic shield, not a curse, just subtle shifts in a never-ending negotiation between human bodies and the microscopic world that surrounds them. And those traces hint at something else as well. The Denisovans themselves were not a single uniform population facing a single uniform disease landscape. Their teeth, their jaws, and the scattered clues they left behind suggest a wider, more varied Denisovan world, one whose diversity we're only beginning to glimpse. For a long time, that wider world was almost invisible. We've been talking about Denisovan DNA, weaving through billions of modern genomes, percentages in Tibet, traces in the Philippines, haplotypes scattered across Asia and Oceania like ghost footprints. But when you turn from DNA to the actual bones and teeth of the people who left it, the scene goes strangely quiet. For Neanderthals, there are dozens of skeletons and partial skeletons, enough to build museum displays, to reconstruct faces, to argue about posture and gait. For Denisovans, we started with a pinky, that tiny fragment from Denisova cave, plus a few enormous molars from the same site, and for years that was essentially it. A handful of pieces from one cave in the Altai Mountains trying to stand in for a population that clearly ranged across half a continent. It's an odd imbalance. On one side, strong genetic signals spread over thousands of miles. On the other, a fossil record so thin you could almost miss it in a drawer. That mismatch is part of why Denisovans feel so ghostly. We know they were there. We can measure their contribution in modern people to decimal places. But if you ask what they looked like, how tall they were, how their faces were shaped, we're still mostly guessing. So paleoanthropologists went looking for other clues. The first came from those teeth in Denisova cave. They're not just big, they're strange. Compared to both Neanderthals and early modern humans, Denisovan molars are massive with complex crowns and unusually tall roots. They don't fit neatly into the known variation of either group. Teeth are durable. Enamel is the hardest substance in the human body. It survives where delicate bones crumble, and teeth carry a lot of information about diet, about chewing forces, about evolutionary history. So when researchers looked at those Denisova cave molars, they weren't just seeing large, they were seeing a distinctive dental pattern, something that might act like a signature. Once you have a signature, you can start leafing back through the fossil record. In East Asia, there are a number of finds that don't look quite like classic Neanderthals or classic early Homo sapiens. Isolated teeth, fragmentary jaws, partial skulls. For decades, many of these were filed under broad labels, archaic humans or regional variants, or just left as question marks. Now, with Denisovan DNA in hand, some of those question marks are being revisited. One of the most famous is the Xiahi mandible, a heavy lower jaw found on the Tibetan plateau and kept for years in a monastery. It's thick, with robust bone and large molars. It doesn't have the chin typical of modern humans. It doesn't slot comfortably into Neanderthal variation either. When scientists tried to extract DNA from it, the high altitude conditions and age worked against them. No usable ancient genome emerged, but proteins did. Inside the dentine of teeth and the mineral matrix of bone, fragments of ancient proteins can survive long after DNA shreds beyond recognition. These proteins are also encoded by genes, and slight differences in their sequences can carry evolutionary information. Using a field called paleoproteomics, researchers sequenced collagen and other proteins from the Xiahi jaw and compared them to those from known humans, Neanderthals, Denisovans, and great apes. The pattern came out closest to Denisovans. It wasn't as detailed as a full genome, but it was enough to say, This jaw likely belonged to something on the Denisovan branch of the family tree. If that interpretation is right, and not everyone agrees, but the evidence is compelling, it pushes Denisovans up to the high Tibetan plateau at least 160,000 years ago, maybe earlier. Think about what that means. Denisovans, or at least a population closely related to the Denisova cave individuals, weren't just living in a chilly cave in Siberia. They were also thriving at altitudes where the air is thin, winters are brutal, and even walking uphill can leave unadapted lungs burning. It fits with the genetic hints we saw earlier, the high-altitude variants in modern Tibetans with Denisovan-like signatures. But now there is a piece of bone, a literal jaw, anchoring that story to place. Other candidates have appeared as well. There are unusually large, archaic-looking teeth from sites in China that some researchers tentatively link to Denisovans based on their shape and age. There are skull fragments and dental remains from Southeast Asia that don't match Neanderthals or fully modern humans and have been proposed as potential Denisovan material. In a few fortunate cases, tiny scraps of DNA have been coaxed out of these fossils or out of sediments in the caves where they were found. Sometimes the sequences line up neatly with known Denisovan genomes. Other times they're more ambiguous, sitting somewhere in the broader archaic Eurasian cluster. The picture that emerges is not a simple map with sharp borders. It's more like a patchwork of overlapping ranges. Denisovan-related groups in the Altai, Denisovan-related groups on the Tibetan plateau, possibly Denisovan-related groups in parts of East and Southeast Asia, all adapted to slightly different environments, cold continental climates, high-altitude plateaus, perhaps humid forests and coastal zones. That suggests ecological flexibility, a species or a group of closely related populations, comfortable in caves overlooking Siberian valleys and in monasteries' backyards on the edge of the sky, able to eat what was available, big game, smaller animals, plant foods, able to handle the different pathogen landscapes and seasonal rhythms each region imposed. But it also highlights how murky the boundaries are. Teeth and jaws can hint at relationships, but dental shape evolves under both genetic and environmental pressures. Chewing tough foods can change wear patterns. Local diets can drive subtle shifts over time. Two unrelated populations eating similar things might converge on similar-looking molars. Proteins provide more secure clues than shape alone, but they don't carry as much information as full genomes. They can say closely related to this group more easily than they can say definitely this, not that. Ancient DNA, when it can be recovered, offers the sharpest resolution. It can show specific Denisovan haplotypes, reveal interbreeding events, place an individual on a branching tree with some precision. But DNA is fragile. It survives best in cold, dry, stable environments. Much of tropical and temperate Asia is the opposite of that. So every line of evidence has its strengths and its blind spots. Morphology, the shapes of teeth, jaws and skulls, lets us work with older and more degraded fossils. But interpretations can be subjective and influenced by expectations. Proteins extend our reach where DNA fails, especially for Middle Pleistocene bones, but they give a coarser picture. Ancient DNA, when it appears, can be dazzlingly precise, but it only illuminates a tiny fraction of the remains that actually existed. Put them together, and you begin to see not just a single Denisovan type, but a wider Denisovan world. Multiple populations, some more robust, some perhaps more gracile, some adapted to altitude, others to forests or coasts. A web of related groups spreading across Asia, leaving behind a mosaic of teeth, jaws, and genetic echoes. Yet the uncertainties never quite disappear. Was the Xiahe mandible part of the same population as the Denisova cave girl, or a cousin line that had already split off? Do all those big molars in China really belong to Denisovans, or to other archaic humans we haven't yet recognized as distinct? How many separate Denisovan-like groups were there, and how often did they meet and mix with each other and with Neanderthals and early modern humans? Different researchers give different answers. New finds shift opinions. A scrap of DNA from cave sediment can overturn a whole neat story drawn from bone alone. So as you picture Denisovans now, it may help to hold two ideas at once. On one hand, there is the solid reality of tooth enamel, jawbone and protein sequences, telling us that Denisovan-related humans range far beyond a single Siberian cave and handled a surprisingly broad set of environments. On the other hand, there is the fuzziness of categories at this distance in time. The edges between Denisovans, Neanderthals and early modern humans blur, especially in regions where they may all have overlapped, interbred, and shared both genes and tools. Somewhere in that blur, your own ancestors stepped into their world. And those encounters, the meetings of different Denisovan groups, the multiple waves of mixing with our line, would leave patterns in DNA that are only now coming into focus. If Denisovans were scattered across such a wide range of landscapes, it would be strange if they were all exactly the same. The DNA agrees. When geneticists look closely at Denisovan-like segments in modern people, they don't form a single tidy cluster. Instead, they fall into at least two, and probably more, distinct groups. One kind of Denisovan ancestry shows up strongly in people from New Guinea, Australia, and parts of island Southeast Asia. Another kind, slightly different in its patterns, is found more in East Asians, some mainland Southeast Asians, and populations like Tibetans. Those differences are subtle. They're not like comparing humans and chimpanzees. They're like comparing two distant branches on the same side of the family, but they're consistent enough that when you run the numbers, the computer essentially says, these segments probably came from different Denisovan populations at different times. That's the first hint that we're not dealing with one Denisovan group, but several. Think of Asia during the late Pleistocene as a patchwork of Denisovan-related communities. Some lived in the cold, dry northeast, nearer to the Altai and Siberia. Others occupied the high plateaus and mountain valleys of Tibet and western China. Still others ranged through the forests and coasts of Southeast Asia, and perhaps down onto the islands that would eventually become Indonesia and New Guinea. Each of these populations had time to drift, to accumulate its own minor genetic quirks, just as modern human groups in different regions do today. Isolated valleys, mountain ranges, rivers, and simply distance would all slow down gene flow. Over tens or hundreds of thousands of years, distinct Denisovan flavours would emerge. Then our ancestors walked into this mosaic. The genetic evidence suggests that when modern humans began spreading out of Africa and across Eurasia, they didn't just encounter the Denisovans once, at a single place and moment. They met different Denisovan-related groups in different regions, and those meetings left different traces. One wave of mixing seems to have involved an early group of modern humans moving through mainland Asia and into island Southeast Asia. Somewhere along that journey, perhaps in southern China, perhaps further south, these modern humans interbred with a Denisovan population whose descendants' genes are now strongest in New Guinea, Aboriginal Australian, and some Philippine groups. Much later, or perhaps in a different place, another group of modern humans encountered a different Denisovan population. The genetic segments they picked up are not identical to the Oceanian ones. They're related, clearly Denisovan, but shifted enough that they form a separate cluster in statistical analyses. These are the segments that show up most clearly in parts of East Asia and in Tibetans, who also carry that famous high-altitude variant likely borrowed from a Denisovan group already adapted to thin air. So instead of one neat interbreeding event, you have multiple introgression events. Different modern human groups, on different routes, mixing with different Denisovan populations. It's a messy, layered story. And then, over the tens of thousands of years that followed, the signal got scrambled. Human populations don't stay put. After those early encounters, modern humans kept moving, splitting, rejoining, replacing each other. Groups that had Denisovan ancestry migrated into new regions and mixed with groups that had none, or with groups that had picked up Denisovan DNA from a different source. Some populations dwindled or vanished entirely, taking their particular blend of Denisovan genes with them. Others boomed and spread, amplifying their version of Denisovan ancestry over huge areas. Later migrations from within Asia, and from back migrations out of Europe and Central Asia, layered additional genetic influences on top. In some regions, like mainland East Asia, the original Denisovan contribution may have been relatively diluted by subsequent waves of people. In others, like remote island or highland communities, isolation preserved archaic segments that would have been swamped elsewhere. What we see today, those percentages on maps, those clusters of Denisovan-like DNA, is not a simple photograph of what happened. It's more like the sediment at the bottom of a river, layers mixed and reworked by countless currents of migration, isolation, and population turnover. To untangle that, scientists turn to statistics. You can't line up a complete Denisovan family tree from fossils alone. so geneticists recreate parts of it from patterns in living genomes. They use tools with names that sound almost like spells: F-statistics, D-statistics, admixture graphs, hidden Markov models. Underneath the jargon, the idea is straightforward. If two modern populations share an unusual set of genetic variants that are rare or absent elsewhere, that suggests a shared source in the past. If certain stretches of DNA are more similar to Neanderthal or Denisovan sequences than to the average modern human background, that hints at introgression. If those archaic-like segments fall into two or more clear clusters, not just one, that hints at multiple archaic source populations. Sometimes the math reveals something even stranger: ancestry from a group that no longer exists in pure form and hasn't yet been sampled in the fossil record. These are the so-called ghost populations. They're called ghosts not because they're mystical, but because we infer them indirectly. If the observed relationships between modern and archaic genomes can't be explained by known groups alone, the best-fitting model often includes an unknown branch, an archaic population that must have contributed DNA, but that we haven't yet identified in the ground. Some analyses of Denisovan-like DNA suggest there may have been more than one such ghost. Perhaps a deeply divergent Denisovan lineage in Southeast Asia. Perhaps a Denisovan Neanderthal hybrid population in parts of Central Asia. Perhaps archaic humans in Indonesia or the Philippines, who are Denisovan related but distinct from the Altai and Tibetan groups we glimpse today. Not all researchers agree on the details. Different datasets and methods sometimes point to slightly different numbers of pulses or slightly different timings. Some models favour two major Denisovan introgression events, others 3 or more. Some argue for deeply divergent branches, others suggest the differences could be explained by structure within a single, wide-ranging Denisovan metapopulation. What most agree on is that the story is not simple and not finished. Every new ancient genome has the potential to change the plot. The first Denisovan sequence from that pinky in the Altai rewrote our understanding of human diversity in Eurasia. The Xiahe mandible proteins nudged Denisovans up onto the high plateau. Sediment DNA from caves in Tibet, by Shia and others, has filled in more dots on the map. Future finds, maybe a partial skeleton from southern China, maybe DNA from an island cave in Indonesia, maybe another jaw pulled from a monastery drawer, could finally link some of those ghost signals to real named populations. Or they might show that what we call Denisovans today actually hide several branches we'll eventually need to distinguish. For now, when you hear Denisovan DNA in modern people, it may help to picture not a single ancient donor, but a chorus. Different Denisovan populations, singing slightly different genetic melodies, mixing with different waves of early modern humans as they crossed Asia. The echoes of those songs, blurred and remixed by time, are what you carry in your cells tonight, and as our ability to read those echoes improves, the outlines of those long-vanished singers, those diverse Denisovan worlds, will come more sharply into view. To imagine Denisovan life, you have to start with their stage. Ice Age Asia wasn't one single frozen wasteland. It was a continent of contrasts, shifting with every glacial pulse. In the far north, winters would have been long and brutal, with steppe tundra stretching toward the horizon, winds scouring open ground, and only the toughest grasses and shrubs clinging on. Further south, belts of conifer forest thickened, broken by river valleys and patches of open woodland. In East Asia and Southeast Asia, climates swung between cooler, drier periods and warmer, wetter ones, forests advancing and retreating over thousands of years. High above on the Tibetan plateau and along the great mountain systems, thin air and cold nights shaped everything that tried to live there. Denisovans, whatever they looked like, were moving through this restless world. We don't have a Denisovan campsite frozen in time, laid out like a photograph. What we do have are hints, a few tools from Denisova cave in Siberia. A tooth here, a jaw there, sediment DNA clinging to cave floors on the Tibetan plateau, and, spread through modern genomes, adaptive variants that seem to have come from them. The stone tools found in Denisova cave, for example, are not wildly different from those used by Neanderthals in the same region. Scrapers, points, flakes, made with Middle Paleolithic techniques. Some layers even show more advanced blade-like tools that might belong to later modern humans. The problem is, caves get reused. Neanderthals were there, modern humans were there, Denisovans were there. The The tools don't come with name tags, so when archaeologists look at those assemblages, they have to be cautious. It's tempting to say, this elegant tool must be Denisovan, but the evidence rarely lets you be so specific. At best, you can say, during the time Denisovans were present, someone here was knapping stone with considerable skill, using patterns of flaking and planning that fit within the broader toolkit of Pleistocene Eurasia. In other words, Denisovans were probably not technological throwbacks. They were likely using stone, bone, and maybe even organic materials like wood and plant fibres in ways that would look familiar if you've seen Neanderthal or early modern human campsites. Fire, butchered animal bone, carefully chosen raw materials, the ordinary, extraordinary toolkit of Ice Age people. Beyond tools, we infer behaviour from genes. That high-altitude variant shared by Denisovans and modern Tibetans suggests at least some Denisovan groups were long-term residents of cold, low-oxygen plateaus. You don't evolve such a specialised adaptation from a single season's visit. That points to generations of life at altitude. Children born in thin air, lungs and hearts working harder, bodies slowly adjusting across centuries until what was once dangerous became normal. Elsewhere, Denisovan ancestry in modern populations seems to carry signatures related to immune function and metabolism. This hints that other Denisovan groups were tuning themselves to different ecological pressures, pathogens in humid forests, nutritional constraints in colder, more seasonal environments, maybe even toxins in unfamiliar plants. Taken together, these clues sketch a picture of a species, or a cluster of related species, that could occupy a startling range of habitats. From Siberian caves with winter temperatures plunging far below freezing, to temperate valleys, to high plateaus, to coastal and island environments in the south. Not because every Denisovan individual could thrive in all those places, but because the broader Denisovan metapopulation was flexible, with local groups specialising in their own corners of Asia. How did they manage that generation after generation, often in small numbers? Think of scattered refugees, a sheltered river valley in northern China, its microclimate a little milder than the plains around it. A limestone cave on the flank of a Tibetan valley, catching sunlight and trapping a bit of warmth. A forested ridge in Southeast Asia, rich in fruit and small game even when surrounding lowlands dried. In harsh glacial phases, Denisovan groups may have retreated into such pockets, their numbers shrinking, births barely outpacing deaths. When climates relaxed, those same refugees would become launch pads. Young adults would push out, following herds, tracking rivers, exploring newly hospitable ground. Some groups would split, some would vanish, a pattern not so different from what we see in other Ice Age humans. At the edges of their ranges, those exploratory movements would have brought them into contact with others. Sometimes that other would have been just another Denisovan band, speaking a slightly different dialect of the same language, shaped by different local conditions. Sometimes it might have been Neanderthals moving east from Europe and Western Asia, and later, modern humans coming out of Africa and then sweeping through the same landscapes. At these frontiers, you can imagine quiet encounters more often than dramatic clashes. A hunting party catching sight of unfamiliar figures along a valley, cautious watching, perhaps avoidance at first. Then, rarely but importantly, meetings close enough for genes to move, a visitor taken as a mate, a captured adolescent adopted into the group, a few seasons of shared hunting in a particularly rich valley before paths diverged again. Those meetings don't leave clear campfire stories in the ground, but they do leave patterns of DNA in modern people. Those patterns tell us that Denisovans were not sealed off in some distant corner. They were part of a shifting web of populations, sometimes isolated, sometimes connected, living mostly in the background of prehistory while more visible Neanderthals and later, modern humans left clearer archaeological signatures. There's a temptation to fill in the blanks with vivid detail, to assign Denisovans a particular kind of clothing, a trademark style of dwelling, a unique artistic tradition. For now, the evidence doesn't let us do that honestly. We can say they almost certainly used fire, stone tools, and complex social behaviours because these are shared across other Pleistocene humans. We can say they were adaptable because their genetic legacy shows adaptation to multiple environments. We can say they occupied refuges, expanded when conditions allowed, and sometimes met newcomers at their borders. Beyond that, we're in the realm of possibilities, not certainties. So Denisovans remain, in a very real sense, people of the shadows. Their bones are rare, their tools hard to attribute, their faces unseen. Yet their presence is woven into the fabric of Ice Age Asia, in the caves that hold their DNA, in the genomes of a living people they once met, and in the silent gaps of the archaeological record where their camps must have stood. To learn more about them, scientists have had to get creative, finding ways to trace their movements and habits without the usual comfort of skeletons and clearly labelled artefacts. And that leads to one of the strangest developments in recent archaeology, learning about an ancient people, not from what they left behind in plain sight, but from the faint molecular echoes they left behind in dust and bone fragments. Imagine walking into a cave that held Denisovans for thousands of years and finding almost nothing. No obvious burials, no clear hearth circles, no rows of tools that can be confidently tied to them. Just dust and layered floors of compacted sediment. For a long time, that would have meant the story stopped there. No bones, no faces, no species. But in the last decade or so, something remarkable has changed. Scientists have learned to treat that dust itself as a kind of archive, a quiet, crumbling, unsorted library of DNA. Every living thing sheds. Hair, skin cells, bodily fluids, tiny fragments of tissue. When humans, or animals, or plants move through a cave, they leave microscopic traces behind. Those traces sink into the floor, get mixed into the sediment, and, if conditions are just right, linger there for tens of thousands of years. This is what researchers call environmental DNA, or eDNA. Not DNA taken from a skeleton or a tooth, but from the surrounding environment. Soil, ice, lake mud, cave floors. In places like Denisova Cave, that sediment is layered like a slow-motion time stack. Year after year, century after century, dust, ash, pollen, animal droppings and microscopic bits of skin and hair settle and get pressed together. To the naked eye, it just looks like bands of brown and tan earth. Under a geneticist's lens, it can become a record of who passed through. The process of reading that record is delicate. First, archaeologists take carefully controlled samples from specific layers of sediment. They work to avoid contamination from modern humans, because a single stray skin cell from an excavator could drown out the faint signals they're looking for. Then, in ultra-clean labs, technicians dissolve those sediment samples, filtering out and concentrating any fragments of DNA trapped inside. What they end up with isn't a neat, complete genome. It's more like a box of paper shreds, tiny broken strands of ancient DNA from many different sources, all mixed together. Animal, plant, bacteria, fungus, human. To make sense of it, scientists use high-throughput sequencing, machines that read millions of DNA fragments at once, letter by letter. Then powerful computers compare those fragments to reference genomes. If a piece matches wolf DNA, it gets sorted into the wolf pile. If it matches mammoth, or cave bear, or birch tree, it goes into those piles. And sometimes, in the middle of that jumble, fragments appear that match ancient human DNA. Some match Neanderthals, some match modern humans, and some, astonishingly, match Denisovans. This means that even when no identifiable Denisovan bone remains in a particular layer, their genetic shadow can still be detected. Maybe a strand of hair fell out as someone slept. Maybe a cut on a hand left a smear of blood on the cave floor. Maybe skin cells were shed as people moved through the entrance day after day, season after season. The material is gone. The molecules linger. In Denisova Cave, this approach has allowed scientists to map out when different human groups were present, layer by layer. Some levels show Neanderthal DNA but no Denisovan signal. Others show Denisovan DNA alone. Still others show both, hinting at periods when the cave's occupation alternated or overlapped between species. On the Tibetan plateau, at high-altitude sites like Baishia Karst Cave, sediment DNA has provided evidence that Denisovans were there long before any clear skeletons turned up. A scrap of jawbone eventually confirmed it, but the cave sediments had already whispered their presence. There is a catch, though. Dirt is messy. Caves are living systems, not perfect filing cabinets. Water can seep through layers. Burrowing animals can churn the sediment. Later visitors can disturb earlier deposits. And DNA itself doesn't stay put forever. It decays, breaks into smaller fragments, and can sometimes move downward or sideways through tiny cracks. So when researchers find Denisovan DNA in a particular layer, they have to ask hard questions. Is this really from the time that layer was formed, or has it drifted from above? Could it be from a single brief visit, or does it represent many generations? How sure can we be about the date we're attaching to this genetic signal? To keep from fooling themselves, scientists lean heavily on stratigraphy and dating. Stratigraphy is the study of those sediment layers, their order, their composition, the way they were laid down. Lower layers are usually older than upper ones, but you need more than that. You look at changes in grain size, signs of flooding, ash from volcanic eruptions that can be correlated with known events, even the microscopic remains of plants and animals. Then there's dating. For cave sediments, this often means techniques like radiocarbon dating of charcoal or bone fragments in the same layer, or other methods such as optically stimulated luminescence, which can estimate when grains of sand were last exposed to sunlight. In some cases, uranium series dating of stalagmites that grew over or under certain layers adds another set of brackets. By weaving all this together, researchers can say, with some confidence, this sediment layer is roughly this old. The Denisovan DNA we find embedded within it most likely comes from that period, not from a later intrusion. It isn't perfect. There are always margins of error. In some sites, the sediment is so mixed that the dates blur into each other and interpretations get fuzzy. But even with those challenges, the payoff is enormous. Without environmental DNA, Denisovans would remain almost entirely confined to a handful of bones, from a single Siberian cave and a jaw from Tibet. With it, the outline of their world begins to widen. We can start asking, where else in Asia might their genetic traces lie, waiting in the dust? In limestone caves in southern China, in rock shelters in Mongolia, in deep dry caverns of Central Asia, in cool shaded overhangs on islands that were once connected by Ice Age land bridges. This is where the future of Denisovan research is heading. Broader sampling, across more sites, more layers, more regions. Smaller and smaller sediment samples, analysed with ever more sensitive methods, able to pull out shorter, more damaged DNA fragments that earlier techniques would have missed. Better reference genomes, not just from one or two Denisovan individuals, but from many, so that subtle regional differences in their DNA can be recognised. With that, scientists hope to see whether there were multiple Denisovan populations with distinct ranges, whether they moved north or south during different glacial cycles, whether they retreated to the same refuges over and over, or constantly shifted their ground. While this work continues in labs and caves around the world, you might find it soothing to know that some of these stories, including the wider saga of bottlenecks and ancient humans, are explored more slowly in book form. If you're curious, the book is available at sleepytimehistory.com, and there's a discount this week for listeners who want to drift through these timelines at their own pace. For now, though, the important thing is this. Denisovans have become one of the first human groups we can trace not just by their bones, but by the molecular dust they left behind. That's a quiet, almost ghostly kind of presence. No dramatic skull on a museum shelf. Just a pattern of A's, C's, G's and T's in cave sediment, telling us that, at some point in the distant past, real people walked, slept, bled and breathed there. As methods sharpen and samples accumulate, those faint patterns will resolve into clearer lines. The map of Denisovan movements will gain new points, new corridors, maybe even surprises that unsettle our current guesses, and each new trace of their DNA, whether in a cave floor or a living genome, pulls their story a little further out of the dark, reminding us that prehistory is still being written, grain by grain, sequence by sequence, in the most unlikely corners of the earth. So what does it actually mean for a living person to carry Denisovan DNA? Not in the abstract, not as a headline about 2% ancient ancestry, but in the quiet, personal sense, in the way it sits inside your cells while you sleep, doing its work without names or labels. One way to think about it is this. Denisovan DNA in modern humans is not a separate layer, stacked on top of something called human. It's part of the same long fabric. A few threads in the weave that happen to be slightly older, with a different journey behind them. When geneticists say that a person in Melanesia might have 3 or 4% Denisovan ancestry, they're not talking about a tiny Denisovan sitting inside a mostly modern human. They're describing a statistical pattern in millions of letters of DNA. Certain stretches resemble what we've reconstructed from Denisovan genomes, more than they resemble typical modern human sequences. Over tens of thousands of years, those stretches have been chopped up. Each time two people have a child, their chromosomes get shuffled. Long blocks of DNA from each parent are broken and recombined. Do this over and over, generation after generation, and any ancient contribution gets sliced finer and finer. This is why most Denisovan DNA in living people doesn't appear as big, continuous chunks. It shows up as many tiny segments, scattered across the genome like grains of coloured sand, a fragment here that helps with oxygen at high altitude, another there that tweaks how the immune system reacts to certain pathogens, many more that, as far as we can tell, are neutral. Passengers riding along, neither clearly harmful nor clearly beneficial. The size of those fragments matters. Longer segments usually mean more recent ancestry. Shorter, more broken pieces point to older events. When researchers look at how Denisovan-like sequences are arranged in modern genomes, they can tell that the main episodes of mixing happened long ago, in the deep past of Asia and the Pacific, not in the last few thousand years. This helps correct one of the common misunderstandings about these numbers. When you see a report that says you're 1% this, 5% that, it's tempting to imagine little purity jars, some part of you really Neanderthal, some part really Denisovan, measured out like ingredients in a recipe. That isn't how biology, or history, works. There are no pure populations in the human story, only lineages that have mixed, separated and remixed over and over. The percentages that show up on ancestry reports are models, built on comparisons with reference genomes and present-day groups. Change the reference or add new ancient DNA, and the numbers can shift. Identity doesn't follow those percentages neatly either. Someone can feel deeply connected to a particular culture or place, regardless of how much genetic similarity they share with ancient people who once lived there. And the reverse is true. You can carry Denisovan DNA in your cells without it saying anything clear about who you are, what language you speak, or how you live your life. Genes are not destiny, and they're not personality tests. They're more like a record of which versions of biological solutions happen to survive. From that perspective, Denisovan DNA becomes less about categories and more about continuity. It's a sign that our ancestors didn't move through an empty world. They met other humans. They formed relationships, however brief or lasting. Children were born, and some of those children carried combinations of genes that let them thrive in new places. Those inherited fragments have taught us a surprising amount about movement, adaptation, and time. Without living Denisovan DNA, we might still imagine that modern humans spread like a clean wave across Asia and the Pacific, replacing everyone before them in a single sweep. Instead, we see something more tangled. In Southeast Asia and Oceania, higher levels of Denisovan-like DNA point to intense contact, maybe multiple encounters with distinct Denisovan groups. In East Asia, different patterns suggest other meetings at other times, with perhaps other branches of the same ancient population. In the highlands of Tibet, a particular Denisovan-derived variant in a gene related to oxygen transport tells a very concrete story. Long before permanent human settlements appeared on the plateau, Denisovans, or a Denisovan-like group, had already adapted to those thin, cold airs. Later, when modern humans arrived and interbred with them, some of those altitude-friendly variants were passed on. Today they help Tibetan populations live and reproduce at elevations that would leave most lowlanders gasping. In island Southeast Asia, Denisovan-related segments are tied to immune functions, hinting at battles fought with unfamiliar parasites and pathogens. Coastal and island environments come with new microbial worlds, waterborne diseases, mosquito-borne fevers, tropical infections. The survival of people in those landscapes may have been shaped in part by genes inherited from ancient neighbours who had already done some of the adapting. Even in places where people today have only a trace of Denisovan ancestry, those traces help map ancient roots. They show that our species didn't simply march out of Africa along a single highway. There were side paths, stopovers, periods of coexistence and exchange. You can think of Denisovan DNA as a set of bookmarks in that long journey, little tags that say, here, in this region, at this approximate time, our ancestors met another branch of the human family, and the meeting left a mark that hasn't entirely faded. At the same time, the presence of these fragments reminds us how much we don't know. We can see the genetic echoes of Denisovans in modern people from the Philippines, New Guinea, Australia, and parts of East Asia. We can detect their DNA in cave sediments from Siberia and Tibet. but the spaces in between are still mostly blank. Were there Denisovan populations in northern China, thriving in river valleys now buried under cities and farmland? Did they follow game herds across the steppes of Central Asia? How far south did they travel along the coasts of what is now Vietnam, Malaysia and Indonesia, when sea levels were lower and the islands were joined? And how many branches of Denisovans were there? The genetic patterns in modern populations hint at more than one, maybe several distinct Denisovan groups, separated by mountains, deserts and ice, each with its own dialects, tool styles and ways of living, all lumped under a single name because we have so little to go on. Beyond Denisovans, the question opens wider. If the DNA of a relatively elusive group like this can still be found in billions of people, what about other, as yet unnamed humans? Geneticists sometimes talk about ghost lineages, populations we haven't identified in the fossil record, but whose DNA seems to have slipped into ours somewhere along the way. Perhaps there were other small local human groups, tucked into forgotten refuges, who contributed a few useful genes and then vanished without a trace of bone or tool. Perhaps some of the adaptations we take for granted today, in diet or disease resistance or brain development, are gifts from those ghost histories. To carry Denisovan DNA, then, is to carry a reminder that your body is a living archive, not of separate sealed boxes of ancestry, but of countless meetings, crises and recoveries stitched together over hundreds of thousands of years. Most of that story will never be fully known. The specific faces, the words spoken beside ancient fires, the details of every journey are gone. What remains are patterns in molecules, hints in cave dust and a handful of bones. But as long as there are new genomes to compare, new sediments to test, the outline of that story can sharpen. Each Denisovan fragment in each living person is an invitation to keep asking, who else was here, walking these same valleys and shorelines before us? What other human worlds rose and fell, leaving only faint chemical traces behind? And how many more of those worlds are still hidden, quietly woven into the DNA of people who have never heard the name Denisovan at all? So we come back, at the end, to something very small. A sliver of bone found in the dark of a Siberian cave. A few grains of sediment scraped from the floor. Chemical traces that, at first glance, look like nothing much at all. From that, we've followed a thread that winds through high plateaus and island chains, through ancient forests and vanished coastlines, all the way into the bloodstreams and breath of billions of living people. We've watched that fragment turn into a genome, that genome turn into a map, and that map turn into a new way of seeing what it means to be human. Denisovans began, for us, as a name on a lab report, an unexpected genetic signal that didn't match Neanderthals, didn't match modern humans, but clearly belonged to something deeply related to both. From there, the story spread outward, to a girl with a Denisovan father and a Neanderthal mother, whose bones told us that these ancient humans weren't separated by hard, clean lines, to a high-altitude gene in Tibetans that whispers of long-ago lives spent in thin, cold air, to immune variants in island populations that hint at unseen epidemics in tropical rains. Now, instead of a single cave in Siberia, we see a whole ghost continent of Denisovan worlds. Many of them are still only outlines. Some may never be more than that. But the faint echoes of their existence are still moving quietly through living genomes every time a heart beats, every time a child is born. And yet, for all that, the ending we can see is strangely quiet. There is no record of a final battle where Denisovans fell, no clear evidence of a last refuge, a last campfire. The fossil trail doesn't narrow to a single dramatic point. It just thins, and then it stops. Somewhere between about 50,000 and 30,000 years ago, the direct Denisovan line disappears from the physical record. Modern humans continue. Neanderthals vanish from their last pockets in Europe and Western Asia. Climate shifts, ecosystems rearrange themselves, coastlines redraw. What happened to the Denisovans in all that? We don't know, not really. Maybe their numbers were already low, their populations scattered and fragile when modern humans arrived in force. Maybe climate swings sliced their habitats into smaller and smaller pieces until there simply weren't enough of them left. Maybe, in some regions, they didn't exactly vanish at all, but were absorbed. Not conquered, not replaced in a single moment, but gradually folded into incoming groups until the distinction blurred beyond recognition. At some point, what we call Denisovan and what we call modern human would have become less a matter of biology than of where you drew an invisible line. The genetic traces in living people suggest that the end wasn't one event, but many small endings spread across thousands of years, local groups going quiet, one by one, until only their DNA remained, tucked into the genomes of their descendants who no longer carried their name. Future discoveries could rearrange this picture completely. Another cave on another mountain might yield a trove of Denisovan bones with dates far younger than any we know now. A single, well-preserved skull from China or Southeast Asia could change how we imagine their faces, their bodies, their range. A new ancient genome from a region we haven't sampled yet might reveal yet another Denisovan branch, different from the Siberian and Tibetan ones, with its own history of meetings and mixtures. If that happened, our maps of Denisovan ancestry and living people would have to be redrawn. Percentages would shift. The where and when of ancient encounters would be rewritten. Even the idea of who counts as Denisovan could change. As more ancient DNA is sequenced, some lineages we now group under that name might turn out to be only cousins. Others, currently invisible, could emerge from the noise of our data like coastlines from a fog. For now, we live with partial answers. Fragmentary bones. Half-lit genealogies traced through molecules. A big, complicated question mark buried inside each of us. If you've listened this far, you've been walking alongside that question for a while, letting it hum quietly in the background as you rest. If you'd like more journeys like this, it truly helps if you gently tap like and subscribe to the channel so you don't miss future stories. If you feel awake enough, you can leave a comment and tell us where you're listening from, or drifting off to sleep. It's always a strangely grounding thing to see all the places this one story reaches. If this kind of deep prehistoric mystery settles well with you, there's also a book that goes further into these hidden worlds and the other forgotten branches of our past. You can read more about it, and if you like, pick up a copy at sleepytimehistory.com. As you lie there, with your own ancient genes quietly doing their work, you might let one last question float at the edge of your thoughts. If so much of our shared human story has been recovered from a single shard of bone in a single cave, how many other worlds, how many other names and faces are still waiting undiscovered in the dark, quietly written into the DNA of people who may never know they carry them at all?